thank you, Jackie, so much, and I'd like to thank you and Rudy and Tyler for including me in this list of speakers. This is going to be a great day, I think, and I'll try to give it some sort of a send-off. For me, a grizzled veteran, bloodied but not bowed, hopefully, in this field, particularly with respect to cancer, I'm going to do something a little unusual. I'm going to really stress translational work that we're excited about today as an integrative activity between the lab and the clinic. I'm also going to stress interactions with the immune system, which is really dangerous because I am in no way, shape, or form an immunologist. My collaborator, Drew Pardole, says that I might have opened the door and someday could be a card-carrying member. This all said, I'm going to stress therapeutic implications of the work we do, and I want to put this in a setting. This is a highly collaborative venture called the Stand Up to Cancer, for which many of you know Phil Sharp, along with Arnie Levine and their colleagues, have done so much to guide the peer review, which has kept this project intact. It's a project in which we're supposed to bring an idea within a 3-year period to have impact, a true impact in the clinic on the management of cancer. And I direct this with my co-director, Peter Jones, who needs no introduction, at USC, and a list of other institutions and participants. It is highly collaborative. Peter and I now share a student, Jinji Panyan, who's working in his lab. I'm going to be discussing a lot of work today done between my lab and my colleague Cindy Zanow at Hopkins, her postdoc, Wheelie Lee, and my graduate student now doing some postdoc work, Sing Sai, in this. And it's integrated with a clinical trials group at all the institutions, particularly at USC and Hopkins. And I'm going to discuss lung cancer today in trials where Charlie Rudin, Roz Juergens, and Malcolm Brock have had tremendous input. And over the past couple of years, a postdoctoral fellow, John Rangel, who's been instrumental in trial design, but also instrumental in some of the laboratory work that has emerged in the setting of those trials. And I want to mention informatics here. We've had at least 3 principal informaticists to contribute to what I'm going to tell you, Harry Esrawan and Wei Wang, my graduate student at Hopkins, a research associate, Harry, and I don't have a picture, but Alexander Block from Ghent University. With this said, I'm just going to reiterate very quickly what Jackie said. We know that epigenetics is controlled largely in the setting of this elegant structure of the nucleosome. It's a very dynamic dance. When we have DNA methylation, shown here at CPGs on 2 strands, this doesn't work alone. If it functions, for example, in transcriptional repression, it does so according to modifications that are attendant to this on the lysine tails and other marks on the histones that make up the nucleosome. Those, in turn, are probably very fundamental to where we put DNA methylation. The silencing mark, lysine 9 methylation on histone 3 in model organisms to man, is known to have the capacity to induce the DNA methylation machinery. The polycomb mark, you'll probably hear more about this today, silencing lysine 27 methylation on H3, has been implicated in recruitment of abnormal DNA methylation in cancer. And demethylases and methylases that influence these, for instance, the demethylase, which if overexpressed can reduce the active mark, K4 methylation on H3, that is also an incentive, if given permission, to bring in DNA methylation. And this has brought a whole translational industry to trying to consider how can we modify these events for the purposes of management of cancer. How can we influence the enzymes that write the marks, those that take the marks off the erasers, and those that read the marks? And you can apply this to histones. You can apply this to thinking about DNA methylation, where the 3 DNA methyltransferases that are biologically active, DNMTs 1 and 3A and 3B, can be targets. You'll hear some probably about the TET proteins and their relationship to base excision repair DNA damage and scent also. And in the future, we're going to probably see a targeting of the proteins and complexes that move these nucleosomes around, which is critical for the entire process. Now, here's a depiction. This has reached the popular literature. This was in The Economist. It depicts this syringe full of epigenetic therapy going at the DNA, and I presume an angry nucleosome on cancer. I look at this each time I present it. All this has to do is wipe its face, and it looks like the cancer wins. That may be a bad sign. But at least this is a very, very growing popular thought about managing cancer by influencing epigenetic changes. Now, as Jackie mentioned, we're in a very exciting period in cancer when we think about the arena in which we're doing this. Our lab is concentrated on DNA methylation changes in cancer, and those constituted by losses where in many of the regions where DNA methylation ought to be present, and many, many genes which should not be methylated at any time in development in their promoter CpG island-containing arena areas that take on DNA methylation. And as she said, there are now mutations frequently turning up in virtually every cancer type that could influence this process, initiate it, create downstream substrates from the mutation that may be the best target points for considering therapeutic maneuvers. So I want to just show you just from the gain of DNA methylation in normally unmethylated CpG island promoters to mention the scope of this, to introduce it as just one of the changes that we think about. So these are infinium blots from Illumina, which Peter Laird, I think, is going to talk more about. But simply put here, all the probes shown on these blots target CpG-rich areas in promoters of a myriad of genes. And green means lack of methylation, and red means methylation. And what I want to point out here is in these cell culture lines of various types of human cancer, the myriad of red probes here, compared to all these normal tissues here, which range from embryonic stem cells to differentiated epithelial and mesenchymal cells. This is not limited to cell culture. If you take the probes, for example, for colon cancer from here and put them through the Cancer Genome Atlas project, and this would be some 150 to 200 primary colon cancers matched to normals here, first you can see a nice separation, and you see the extent of these hundreds of genes. Now, obviously, all of these genes that take on this change are not drivers. This is one of the big questions in the field. Some are classic tumor suppressor genes where we know this epigenetic change really can be in place of a frank mutation in that gene for loss of function. And I'm going to address the possibility of using this as a therapeutic target, although I'm going to tell you the caveat. Now that we're getting some traction in this with the way we approached it, I'm the first one to say our next job is to figure out what percentage of what we may be achieving is really due to this as the therapeutic target. This will be a challenge. But what we chose to do within the scope of the Stand Up to Cancer project is to take one old drug and one somewhat old but newer drug and combine them in an approach that we thought we could use these drugs in a way they had not been used in the past, particularly the DNA demethylating drug Vidasa, which had been very toxic in the past, so much so that it would have been hard to see that it gained any traction in solid tumors. And use these two drugs with a laboratory paradigm to study how that use might go, combined with clinical trials that were in and around this. And again, now the big challenge, because I'm going to show you some results we're very excited about at an early stage, and we really keep needing to determine what's the mechanism for the clinical efficacy. How did we approach this? Why 5-azacitidine? Well, this DNA demethylating drug, whose activity in that regard was discovered by Peter Jones around 1980, has now been FDA approved for the pre-leukemic syndrome myelodysplasia, and it works quite well in many forms of AML, acute myelogenous leukemia. It's only come in the hematopoietic diseases to have that sort of traction as a remarkable thing in oncology occurred, marked dose reduction. So 10-fold lower than the MTD. And as a result of those low doses, it was powerful enough in MDS that it's been FDA approved. Our question is, could we do this in solid tumors? We took several things into account in our lab work. One, that patients take a long time relatively to respond to these drugs when they have MDS. They may take several months before you actually see therapeutic traction being gained. And it seems to be a non-cytotoxic approach or result in the early parts at least, and suggests that you even could be affecting or exhausting self-renewing cell populations within the tumor. The target for this drug is an interesting one. Most people know it by its blocking of the catalytic activity of all three biologically active DNA methyltransferases. But we are also heavily considering, with more and more laboratory evidence for this, that the transient physical removal of the DNA methyltransferases from the nucleus of the cell with initial exposure of these drugs could play a big effect, because these drugs have experimentally quite fine transcriptional co-repressor functions independent of their catalytic sites. We think this is going to be very important. They can collaborate with histone deacetylases in maintaining silencing of hypermethylated genes, for example, and we know in the lab that inhibition by employing a histone deacetylase inhibitor with inhibiting these can additively re-express genes. And that's a paradigm that led to our use together, although again, we have much to go to figure whether that is the molecular mechanism for some of the things that I'm going to show you. We had a hint of how to use this drug really back as early as 1980 in this experiment by Jones and Taylor, where they found that azacitidine would take immortalized fibroblasts and turn them into muscle cells. And if they plotted the muscle cell differentiation by counting the micronuclei in the blue and matching that to the DNA demethylating effects shown in the red, they found that really it was at the lower doses that you maximized differentiation far before you plateaued out for decreasing the DNA methylation. This, we would purport, is probably a cytotoxic range. This is the range we need to think about in therapy. So the laboratory approach that we've used is to try to set up very transient exposure to leukemia and solid tumor cells with the lowest doses in a 3-day period here that would not cause significant DNA damage, would not cause a lot of cell cycle check or a lot of apoptosis. Finding that dose, which ranges for different tumor types, but always in the nanomolar range, we would then stop the drug, rest the cells, and then equal numbers of viable cells have been put into now many assays between my lab, Cindy Zanow's lab, which include self-renewing assays long-term, xenograft models in the mouse, and more use of primary non-cultured samples to do the same transient exposure and watch the effects in various assays, such as sphere assays, which enrich for more self-renewing cells, or orthotopic implantation. I will tell you we are getting evidence, which is really nice, we think, that we can influence some of these assays even more than those that don't enrich for such cells. So we think that this has the possibility of, quote, reprogramming cancer stem-like or self-renewing cells in addition to the other cells in the tumor. We use this window in here to do a critical thing for us, and that is to do genome-wide timed studies after drug exposure before we put these cells in. So we do DNA-wide methylation assays by the Infinium assay. We do gene re-expression studies by Agilent, and obviously for individual genes, we do validation and a lot of pathway analyses. What have we learned just wholesale about some of those approaches? We know that, for instance, in leukemia cells or in breast cancer cells, nanomolar doses beginning at day 1 and by that end of that 3-day exposure, and as I said, take DNA methyltransferase 1 way down by Western blots of nuclear extracts here. We know that's accompanied by an actual loss of DNA methylation. This peak here would be promoter methylation in this particular leukemia cell culture line, and 10 nanomolar will take it down about 30% overall. And we watch that, and we can see that when we withdraw the drug at day 3, there's a delay and a stepwise return of that methylation back to normal, which can vary for groups of genes. This is probably very important. And these genes can vary in how long they will re-express. The HOXA9 gene on this Agilent array here is not peaking until some 11 days after we withdrew the drug here. This is a death-promoting gene here, and you can validate that. This is the P15 tumor suppressor gene in 2 lines of leukemia here, where you see its peak by RT-PCR is 11 days after we withdrew the drug. So taking all this, I'm going to come back to the lab, but now I want to go for a few minutes to the clinic and tell you about our first efforts in our group in solid tumors to use these low-dose therapies for therapeutic management. And I'm going to be talking about lung cancer. I don't have to remind many of you in the room that advanced non-small cell lung cancer, adenocarcinoma, while we have mutations, maybe 20% of the patients that we can target, we have virtually as an unmet need in advanced squamous cell carcinoma, where there's very little to offer advanced patients with advanced disease. And many of the targeted therapies, while they're initially quite robust, often come back and are quite resistant when they do so. So, in the first 62-some patients that we've treated with a far below dose of the DNA demethylating agent by days, far below the maximal tolerated dose, and similar to what's used in the liquid tumors, even a little lower, with the histone deacetylase inhibitor tinostat in a regimen that's very well tolerated by patients. We've had about 3% or so, minority, but this would be a lot of people with lung cancer who have responded to this therapy alone. When they do, the characteristics are that it's very robust and it's very durable in those patients. So this is one man here, a typical patient with pretreated lung adenocarcinoma, or actually this is squamous carcinoma, I forgot, sorry. And you can see that after 2 months, 6 months, you can see regression of this lesion here, this lesion in the mid-chest here, and liver metastases here, which this disease has stayed away for now up to 4 years. He has some progression here and there, still doing well. And I'm going to come back to him as he plays into the rest of the story in a moment. But what we really began to notice and published in November, but now have extended, is that many of these patients, and these are 28 patients here, who went on to therapies after they progressed through even short courses of this therapy, were showing dramatic responses in the clinic to their subsequent therapy. And so we noticed that less of them were progressing, shown on this waterfall plot here, than were having stable disease or these marked responses of actual tumor response to either chemotherapy or what we recently began to notice, now 5 patients who had received a form of immunotherapy that I'm going to talk about in a moment. So all 5 are getting therapeutic efficacy. The worst is this patient who stabilized for 7 months and has progressed. These 3 patients, over a year now, are still improving. Their RECIST criteria scan responses are getting even better. And this patient's only been on, this is the gentleman I just told you about, after 4 years, having 15 months total of epigenetic therapy, is only 1 month on this immunotherapy and already showing some tumor reduction and stability. So we see therapeutic efficacy in all of these patients, and obviously these are low numbers, but very striking and very exciting to our oncologists, immunologists, and to us. I think most vividly, let me show you the 3 biggest responders, because these scans are ones even I can interpret. Here's the starting lesion after epigenetic therapy was taken away here, and over months, here's the shrinkage with the immunotherapy, and this lesion here too. This next patient has a huge mass right here, and you can see that over the course, now it's been over a year, has reduced that mass. And then this very large mass in the mediastinal region here in this patient, which has disappeared essentially. So that's very vivid. I'll put it in a little perspective. So we've seen about 75% of the patients who've gone on to subsequent therapy, including those with the immunotherapy, who are getting, quote, therapeutic effect. But over 20% of these are actual responders. Only one of the responders that I'm aware of has died. The rest have not. So we don't know median. We don't really have a good handle yet on how long these median responses are going to be. But they're now well out over a year for most of these individuals. So the challenge is obviously in clinical trials we have to get the n-value up, which is what our next huge effort is in the project, with trials to formally test this. But let me tell you a little bit about the immune therapy and come back to the lab and tell you our growing hypothesis at a molecular level of why this immune therapy may be sensitized by the epigenetic, quote, therapy that we had given first. So these 2 papers just came out in the New England Journal and at the recent clinical meetings, ASCO, have attracted great attention. Targeting immunotolerance in cancer by using antibodies against either the PD-1 receptor on the T cells or antibodies against its ligand, PD-L1, which would be expressed on the tumor cells. And so this has gained a lot of excitement. It was particularly exciting that patients with the same type of advanced non-small cell cancer we're talking about, and which included those 5 patients that I just told you, there was about an 18% response in the PD-1 trial, about 10% in PD-L1. So that puts it in a little bit of perspective of this early high percentage response that we're seeing in those 5 individuals. So again, my immunology is I'm learning, But what this therapy does, there are two forms of this therapy. One that's approved by the FDA for more hematologic tumors, CTLA-4, an antibody against that receptor, and the one being used here, PD-1 and PD-L1. But both of these drive this process of tolerance. This tolerance could come about in at least one of two ways. Either a tumor cell in its chronic interaction with the T cell, that T cell can stimulate right here after MHC antigens interact with the T cell receptor. That chronic state can stimulate that T cell through interferon gamma and the STAT pathway to actually begin to produce PD-L1, or oncogenic signals, and we think this is very important. Various oncogenic pathways are now being more and more recognized can upregulate PD-L1 as well. Of the 25 patients in those trials I showed you, early result that's important is none of the 17 that lacked PD-L1 on the surface of the cell responded. Early data from the group. So let me come back to the lab. What we've been able to do is take among our cell lines in that paradigm that I showed you for transient exposure, and these are the mouse readouts. We've now worked with some 15, but these are 8 lines of non-small cell lung cancer on which we have the genomic data. It's somewhat paralleling the clinic in that 4 of these 8 lines don't respond at all. And interesting, early on, these are lines that either have KRAS mutations or EGFR mutations, and we'll see how that holds. 2 were intermediate. 1, importantly, is a squamous, and only 2 had this marked delay in latency in the mouse after the initial drug exposure of some 6 weeks to maybe 5 weeks. So somewhat reminiscent of what we'd see in the clinic, and I'm going to be talking about the phenotypes of these lines in the context of the molecular data that I'm going to end up with. I'm going to show you first what we've seen in globally interpreting all the genomics that we have done so far in the pathways, because what has really been most interesting to us is multiple transcript changes that would upregulate either increased production of the MHC1, the critical molecule on the surface, including transcripts, and the caveat is we're doing proteins on all these now. This is transcript data. On MHC1 assembly, it's processing in the endoplasmic reticulum and anchoring on the cell surface. And really critical, we've seen big responses in pro-inflammatory responses, which are closely linked to viral defense-like responses on the part of the cells. And what's interesting to us is this could drive immunity, but each of these is also clearly attached to pathways that can do the host cell in, autophagy and apoptosis, and that gets somewhat important to the phenotype of these cell lines when put into an immunoincompetent mouse as these changes aggregate with anti-tumor responses even there. And we've seen evidence that this can actually drive tolerance, which would set up the target. In and of itself wouldn't be a good thing alone. And that the balance between these and other anti-tumor effects we see at the transcript level determines the phenotype. So let me just quickly end by carrying you through. I'm not going to regale you, and I can't show you all the transcripts, but I want to show you some actual data that we see here. So this is for the assembly of MHC1, which obviously starts with T-cell receptor interaction to stimulate its production, goes through a series of proteasomal degradation steps, ending up being anchored into the endoplasmic reticulum by this TAP1 and TAP2 proteins here, and then with a participation of beta-2 microglobulin and another protein on the surface. So we've seen upregulations of transcripts for virtually all of these. So the 2 dark blue lines here are the 2 most sensitive lines. The squamous lines, number 4 here, and the intermediates are the light blue, and all of these are the ones that don't show that response in the mouse that I showed you. Each point here, this is day 3 when drug is stopped. This is 7 days following withdrawal, and the point is always the signal after drug over the MOC for that point. And what you can see here is the marked upregulation of each of these HLA antigens at the transcript level in these cells, and that goes through virtually most of them except this placental-produced HLA-G here. 2 key molecules which can be mutated, Ricardo Della Favara reported 15% mutation of these genes that help anchor and assemble on the surface, beta-2 microglobulin CD58, quite prominently upregulated, particularly again in the sensitive lines, even in the immunoincompetent mouse. Here are the proteins for proteasomal degradation in the endoplasmic reticulum, which helps hone the assembly of the HLAs, LMP2 and 7, and here's that anchoring protein in the endoplasmic reticulum TAP2. PD-L1 itself, and this is we think is important if the proteins follow, is upregulated in multiple of the cell lines. Again, tends to go up. This is that squamous cell line. The interesting thing is that's in contrast to what we see for the alternative ligand, PD-L2, or sort of the CTLA-4, which I mentioned, molecule, the ligand. We've seen no change in that. As I said, interferon gamma is important to this PD-L1 production by driving STAT. And here, as opposed to alpha and beta receptor for interferon, we see this marked increase in the transcripts for the interferon gamma receptor. These signals are interpreted by transmembrane proteins and this viral defense system, which has 3 stages, one of which a protein called IFI-27, which is upregulated, can actually trigger death in the later changes, and what Flavell has called this inflammasome, where many death domain proteins are upregulated. And I'll just show you examples. STING starts the process of viral recognition of double-stranded RNA. It's a helicase and works with several family members shown here, including a protein, if it goes down, which allows apoptosis. And so here is STING itself, and here is 1, 2, 3 of those family members here, and that's that squamous cell line, which is of great interest to us. And I'm about to finish here. And this is the downregulation of that protein that would allow apoptosis in the most sensitive line and that squamous line. And finally, other death events. This is a protein that Peter Jones is actually functionally testing in colon cancer cells when demethylating goes up, is a repressor of survivin. And here we see at the late point this marked downregulation in survivin in the most sensitive line, which encompasses downregulation of the survival protein BCL2. We've actually begun to place a score on this. This is a gene enrichment score for all these genes. These are the most sensitive lines here. This would be the 2 most sensitive, and you can see this score which aggregates towards the most sensitive lines in the immunocompetent mouse. Finally, we've been able to get some early thought about how this plays out in primary cancer by use of the Cancer Genome Atlas Project to look at basal values of these genes for RNA-seq. And what we see is, consistent with a paradigm of immune evasion, that in both the adeno and squamous, many of these genes which would account for immune evasion have very significant downregulation in the tumors basally compared to normal. And when you go to heat maps across adeno and lung in the squamous, in the TCGA, you see normals separate away from the tumors, but we have this large group of tumors here with the lowest regulation of these genes basically, including a big group of squamous cell tumors here. So we may get groupings that'll help us understand. So this is the paradigm again. We're upregulating immune responses, death responses, but also upregulating some facets of the targeting that might present itself and help explain why breaking immunotolerance would help this so much. I thank you very much. I hope I'm